So this is a video on the cell-mediated immune response in specific immunity. Now, it's the second version of this. The first one, called cell-mediated, I actually recorded for a different course, and it uses the slides from a different textbook. So I'm going to re-record or record a different version of it using the slides from your textbook. Factually, they're the same, so either one or both, you might benefit from seeing it a different way. So, so here we go. The cell-mediated response is really a product of T lymphocytes. The T lymphocytes are what does it. And there's a couple of different types of T lymphocytes. There's probably about 4 or 5, but there's 2 that specifically we're going to talk about. And they have different types of receptors on their surfaces. They are still antigen receptors, but we call them CD4s and CD8s, and this determines on what their job is in the cell-mediated response, and this will become clear later. But basically, they become helper T cells or killer T cells. Helper T cells help, and killer T cells kill. So the CD4s, the ones that have the CD4 receptor, become helper, and the CD8s become killers, cytotoxic. They destroy cells that are harboring foreign antigens. So really, there are like, these are like specific natural killer cells. Now there's other types. There's regulatory. So these regulatory T cells are suppressors and enhancers and things like that. And then there's the memory T cells that come after clonal selection. But nevertheless, the 2 major types, helpers and killers, CD4s, CD8s. So you get an immature lymphocyte in the bone marrow. It migrates to the thymus, where it matures, gets its receptors. If it gets a CD4 receptor, it is destined to become a helper T cell. If it gets a CD8 receptor, it is destined to become a killer T cell. So what happens is then they leave the thymus and they migrate out into lymph nodes or spleens or any lymphatic tissue, where they sit and they wait, being exposed to antigens. Now, they can't be exposed to antigens on their own. They have to be processed to major histocompatibility complexes, so they need processed antigens to become activated. Now, the helper T cells get activated by a class 2 MHC in an antigen-presenting cell, and when it gets activated, it immediately clones, makes memory cells, and cells that are used. Okay? So it's, again, this clonal selection, the same as in the B lymphocytes. If it's a CD8, it gets activated by an antigen-presenting cell, but the class 1 MHC. And when that becomes activated, it clones. Some of them become memory cells, and some of them go on to become killer T cells, cytotoxic T cells. So it's the same sort of process. The difference is that the activation is in the MHCs, the class 2s and the class 1 MHCs, rather than the antigen right on the pathogen. Right? So in humoral, the antibodies target things that are in extracellular environment, in either secretions, mucus and things outside of the body, or fluids within the body, blood, lymph, interstitial fluid, that kind of thing. And they attack the pathogens themselves. In the cell-mediated response, they only will respond to processed fragments of the antigen on the surface of one of our own cells. So it's got to be incorporated into an MHC. And the targets of this are our own cells that are infected by either virus or bacteria, or abnormal cancerous cells, or cells, body cells that just aren't supposed to be there, like grafts, kidneys and livers and things that are donated. So transplants will do it. Because those are really our body cells that are displaying the wrong major histocompatibility complex. So because they kill our own cells, that's an expensive proposition to the body, and we don't want that just to happen lightly. So the activation is a little more complex. So the T cell has to recognize that the antigen is non-self. It comes from a pathogen or something foreign. But then it also has to recognize that the cell that is involved with presenting that antigen is a cell. So you need this major histocompatibility complex protein. So you need simultaneously to recognize that, okay, this is our own tissue and there's something wrong with it. So this happens. So the class 2 only is the antigen presenting cells. The class 1 is any infected cells except for red blood cells. So what happens in the class 1 is antigens that are made within the cell are migrated out to the outside and become part of the kind of the flag, as it were. And it can get complicated, but we're not that worried about it. Basically, the antigens end up in the cell and get incorporated into the cell membrane. And we'll just leave it at that. So these are all the steps of it. And I'm not going to ask you to memorize these steps. In the class 2, the exogenous antigens, so the antigens from the outside in the body that was phagocytized becomes incorporated. And I don't want you to know that in any more detail than it incorporates this antigenic peptide from the outside, from an exogenous source. So, to activate this, you need to have the cell come into contact with an antigen presenting cell who's presenting fragments of the antigen on their major histocompatibility complex. and the T cell has to bind with it. But then the T cell has to get verification that it's doing the right thing. So it needs to be co-stimulated. It almost needs to get permission. Now it happens in a couple of different ways. So the receptor, the CD4 and CD8, bind to the major histocompatibility proteins, right? CD4s to class 2, CD8s to the class 1s. But that binding takes place nevertheless. Right? And the antigen is incorporated because of phagocytosis. The big part of this is that after it's bound, you end up with a co-stimulation. The picture makes more sense. So the antigen is brought into the dendritic cell, which is a phagocyte. It's incorporated into its and displayed in its MHC. The immunocompetent CD4 binds with it, and then there's co-stimulation happens across here. You end up with cloning, and you get a whole bunch of helper T cells from this. So this becomes the helper T cell. And what's interesting is that the helper T cell can be co-stimulated simultaneously from the same antigen presenting cell, because the CD4 is capable of doing that. And it does it with so the T cells are co-stimulated by the antigen presenting cell. The helper Ts are, at least. And they do it with either proteins that are on the antigen presenting cell or cytokines. Cytokines means cell movers. They're like local hormones almost, interleukins between white cells. So you need to have this co-stimulation to have it happen. If you don't get co-stimulation, then the T cells stop reacting to the antigen. They, it's almost like it gets used to it and stops seeing that antigen as something to attack. So you don't want that to happen. When they are activated, we get cloning. They proliferate, they clone, and they start to do what they're supposed to do. So if it's a helper T cell, then they'll do that. If it's a killer T cell, then they will start looking for things, tissue cells to kill. This, on the first time that these are activated to that antigen, it takes about a week. And the T cells, once they do their job and once the antigens are no longer there, then the T cells start to die automatically. What you don't want is the T cells to be around too long, because they might start killing other tissue cells. And like I said, their job is really expensive to the body. I'm not going to worry about the different types of interleukins and the cytokines that are used to co-stimulate. I mean, if you're interested in it, we can sit down and chat kind of one-on-one about some of this stuff. It's interesting when you start to do research, cancer research and that kind of thing, looks at the specific. This is how the new cancer treatments are targeting specific things, but nevertheless, what you want to happen is the T cells to be co-stimulated and do their job. So what do the helper T cells do? They really activate T and B cells. So here's the thing. We activate a helper T, and then those helper Ts will activate a naive B cell and start the humoral process against that antigen. So they can almost act like an antigen presenting cell to the B cell, sort of thing. They will activate killer Ts to that antigen as well. They cause the T and B cells to proliferate, and they activate macrophages. They stir up the macrophages and get them eating more. And so then these end up with chemotaxis and all of that stuff. So that whole non-specific response gets enhanced by the helper Ts. Without the helper Ts, there really isn't very much going to go on. They're central to the role of it. So this just is saying all the same thing. So they stimulate B cells. Like B cells can be activated without the helper Ts by the independent antigens. But a lot of antigens, especially any antigen that is within a cell, needs to be stimulated by the helper T cells. The B cells need to be, and so that the B cells are primed and the antibodies are ready when the killer T liberates those pathogens from the infected body cell. So they got to kind of be ready. Yeah, so this is how they do it to the B cells. So they activate that way. B cells don't really need to be co-stimulated. Right? The helper T cells cause the dendritic cells to express co-stimulatory molecules, and so it's a bit self-fulfilling. So what do the cytotoxic T cells do? They kill other cells. They're activated, and then they're in the blood and their lymph, in the lymphatic tissue, but they don't work in a lymph node the same way as the B cells, because you don't bring the pathogens to them. They have to go to the tissues that have the infection. So they circulate. And when they latch on to the antigen that they are programmed for. So they have to go to the site where a lot of the others, the pathogen comes to them. Right? So the main target is virus-infected cells, or cells that parasites, intercellular bacteria go. Cancer cells are really important, and foreign cells. So these are transplant cells. Okay, so they bind to any MHC, non-self complex. So an MHC that has the antigen incorporated in it, the non-self incorporated into it, and they destroy that cell. And they do it by releasing perforins, which poke holes, and enzymes that cause the cell to die. But it's expensive. I've said this a number of times. You don't want to kill your own cells unless they are co-stimulated. And the helper Ts are what do the co-stimulating to the killer Ts. Now the NK cells are a type of killer T cell, except they don't have the class 1 major histocompatibility complex. So they do much the same thing, but they're not specific. Just for completeness, we'll talk about the regulatory cells. So these they inhibit, they turn off the immune response, and so they're important in that regard. So when you look at it, this is the whole thing. An antigen on a pathogen usually triggers the innate defenses, so the surface barriers keep it out, the internal defenses, so this would be things like fever, inflammation, cells like phagocytes, natural killer cells. This would be things like interferon and complement, all get activated innately. The antigens get engulfed by a dendritic cell or a macrophage, which becomes an antigen presenting cell, which then activates, we'll go over here first, the naive CD4, which then becomes an activated helper. There's some memory, so we clone and we make a lot, right? The activated helper will activate the B cell that will clone and give rise to cells that become plasma cells and cells that become memory cells, which then make the antibodies, which neutralize, precipitate, agglutinate, and activate complement, which goes down here, which makes it easier for the macrophages to eat and destroy the intruder. If the intruder gets into cells, then the antigen presenting cell activates the killer T, the naive killer T, which is co-stimulated by the helper T, which clones, you have memory cells, and then activated cytotoxic, which kills the cells that the intruders are hiding in. The intruders then end up in the interstitium where they're eaten by macrophages. The helper Ts also stimulate the NK cells and rear up and get the macrophages working more, which then eat and kill and do their thing, and somehow comes back to inhibit the intruder. So all of these things work together, and you'll notice that the helper T is central to all of this. So this leads us to the idea of transplants, and there's 4 ways that you can transplant. Autografts is from within one person, and these are the best. So these are like skin grafts and things like that, where they take some skin off your bum and they put it over a non-healing wound or something like that. And because the MHCs are identical, then there's no possibility for rejection. Isografts are the next best thing. That's between identical twins, but not all of us have identical twins. Allografts are probably the most common. So this is between individuals. So these are the kidney transplants and things like that. This is liver transplants, heart transplants. Really, it's also blood transfusions are considered allografts as well. Now, xenografts is if you're grafting something from a different animal species. Probably the most common thing is heart valves from a pig heart. But they have transplanted baboon hearts into babies. They've done all kinds of xenografts. There is a real moral and ethical thing about that, but whatever. I think if you're choosing between dying or the ethics of using another species' organs, you'll choose the other species' organ. So rejection happens because the immune system attacks it. So really what you need to do is do immunosuppressive therapy. So the corticosteroids, immunosuppressant drugs, anti-proliferative drugs, are all important to stop this specific response about attacking the grafted organ. They all have problems. You become much more prone to infection, more prone to cancer, to that kind of thing, because you're immunosuppressed.